Good afternoon. This is the next lesson in the Inheritance and Evolution Notes booklet on sex linkage, or firstly sex determination, and then genes which are linked to the sex chromosomes, so the X and Y chromosomes in humans. So the first thing we'll do is look at how sex is determined, and then use some genetic diagrams to show how sex-linked conditions are inherited. Okay, so what we have here is a karyotype. And a karyotype shows all of the chromosomes in a human somatic cell, so in a human body cell. And we can see that we've got chromosome number 1 down to number 22, and then the 23rd pair of chromosomes here is XY. And that would mean that this individual with a karyotype, such as this one, was male. Okay, because they possess an X and a Y chromosome. Now if we just look carefully at that X and Y chromosome, we'll notice that the X chromosome is much larger. And in fact, the Y chromosome is missing, essentially, this part of the X chromosome. Over millions of years, that region of the Y chromosome has been lost. And we'll come back to why that is important later on. Okay, so if we move on to those sex chromosomes, so the Y chromosomes are very small. They contain just a few genes. I've got 78 genes here, there may be a few more than that. Whereas the X chromosomes are much larger and contain a lot more genes. So, because females can only produce X gametes, i.e. a gamete which contains an X chromosome, because obviously a female is XX, after meiosis, those X chromosomes will be separated into the gametes, like so. Okay, so each of the gametes will contain an X chromosome. So it is the sperm, really, then, that determines the sex of the offspring after fertilisation. Because the sperm are XY. After meiosis, the gametes will contain either an X chromosome or a Y chromosome. So if we were to produce a genetic cross diagram to show sex determination, it may look something like this. So, female gametes, and then the male gametes in our Punnett square. Oops, very good line there. And we see XX for female, XX for another female, XY for a male, XY for a male. So I doubt this is news to anyone, but there is a half chance, or 0.5 chance of it being male, and the same for female. Okay, I doubt you'd ever be asked that in the exam, but often what you might need to do is look at the stem of the question where it may mention male or it may mention female, and both of those things are phenotypes. Okay, so if a question asks for what is the probability of a male with a particular condition, well, obviously half of the offspring can only be male. And we'll come back to look at an example of that later on. Okay, this is another diagram showing the same thing I've just shown before. So gametes produced, X and Y in different gametes in a male, X and X in the female, There's the Punnett square, 50% chance of male, 50% chance of female. Okay, so sex linkage then. This is where the gene is carried on one of the sex chromosomes. Okay, now as the X chromosome is so much larger than the Y chromosome, it's likely, and in fact all of the examples that will be used on the A-level exams, those genes will be linked to the X chromosome. So we can think of sex linkage as X-linked as well. Okay, use the term sex linkage, but we will only ever consider X-linked chromosomes. So the X chromosome has what's called a non-homologous portion. If we go back to the carrier type here, if this is the homologous portion on the X and Y, this blue part that I showed earlier on, this is that non-homologous. So non-homologous portion. So that non-homologous portion, if there are genes with a loci in that non-homologous portion, well, they would be present on the X chromosome, but not present on the Y chromosome. Okay, so men will only have one allele for some of those genes on the sex chromosomes, i.e. a man is XY. Oops, that wasn't a very good Y, as it looks a bit like another X. XY. If that gene is present on this, where is it? On this non-homologous portion, that gene will only be present on the X chromosome. And to show that, we use a symbol in superscript, or a letter in superscript, to show that that gene is present on the X chromosome. So males, therefore, will always show the characteristic when they have only one copy of the gene. And that doesn't matter if it's dominant or recessive, because they've only got one copy of that gene, so one copy of the allele. If that allele is dominant, they will show the dominant phenotype. If that allele is recessive, they will show the recessive phenotype. Okay, but they only have one copy of that allele, so they will always show the sex-linked phenotype. Heterozygous, another thing to consider here, is heterozygous females for a recessive sex-linked feature are carriers. So heterozygous meaning they have two copies, so maybe they are using that same letter to represent the allele. Now, because of the because this sex-linked condition here that we're considering is recessive, the females have that recessive allele, but that is masked by the dominant allele. But in this case, with sex linkage, we refer to the females, if they are heterozygous, as being carriers. So you need to look for this word in the question. So carriers and heterozygous females essentially mean the same thing. And it is a clue in a question that you need to underline. If you see this term carriers, you need to think, okay, that means it must be heterozygous. Okay, some examples of sex-linked conditions include colour blindness, haemophilia, and muscular dystrophy. And we'll have a look at a couple of those examples shortly. There are others. In your question booklet, there are quite a few different examples. And I will warn you that the examiners absolutely love sex linkage. And I think nearly all of the genetic cross diagrams that have been present on the most recent paper 2 exams have involved some form of sex linkage. What they really love, like the gold standard, is what we refer to as sex-linked dihybrid crosses. And yeah, like I say, this is a warning. The examiners absolutely love these. And we'll do some examples of that later. Not on this video, but I'll do it on a different video. Okay, so how are sex-linked diseases inherited? You've probably gathered already that a male can only donate an X or a Y to the offspring. Females can donate 2 X's. So what we're saying here is a male could only pass on his Y chromosome to a male offspring, to a son, whereas a female would pass on the X chromosome to a son or the daughter. So males must therefore receive their Y chromosome from their father. Okay, and this is a really important thing to consider, because that means, if we remember, that those sex-linked diseases, most of which are going to be, in fact, all of those sex-linked diseases will be X-linked. So they're not passed down from the father. Their X-linked chromosome and the sex-linked disease allele, therefore, must come from the mother. This means that if the mother does not suffer from the disease, but the offspring, a male offspring, shows the phenotype of that disease, they must have received it from a heterozygous mother or a carrier. So males suffering from a sex-linked genetic disease are able to pass on their allele to their daughters via the X chromosome, who would then become carriers of the disease, assuming that the mother wasn't or didn't show the phenotype for that particular disease. Now we'll have a look at some examples to demonstrate this. The first of which is colour blindness. So colour blindness is an example of a sex-linked condition. Okay, the allele that causes colour blindness is recessive, and it is present on the X chromosome. So I use the symbol R there. Okay, so a male who carries that recessive allele would therefore be colourblind. A female who has just one copy of that recessive allele on their X chromosome, on one of their X chromosomes, would be a carrier. But phenotypically, she would have normal colour vision. Okay, but therefore she goes on to have offspring, give that X chromosome here to a son. And if she did so, that son would then be colourblind. So, red-green colourblindness is a sex-linked condition. As we said before, it's recessive. It's got a gene loci on the non-homologous region of the X chromosome. So we went right back to the start. Remember, there is that non-homologous region, or the non-homologous portion of the X chromosome. Therefore, it is much more common in males than it is in females. And the reason for that is the females would require 2 recessive alleles, i.e. that, whereas a male only requires 1 recessive allele, i.e. that. So, males therefore always inherit the colourblind allele from their mothers. Males can pass on the colourblindness allele to their daughters, who would be carriers if they are heterozygous. Sorry, that should be a heterozygous genotype rather than phenotype. So if they've got this genotype, no they aren't. If they have this genotype, they would be heterozygous and carriers. So males cannot pass on colourblindness to their sons, because obviously their sons would receive the Y chromosome, and that has that doesn't have the homologous portion that's present on the X. So let's have a look at inheritance of this then. The genotype of a son who is colourblind must be, I'm using B's now rather than R's, X with a lowercase b, Y, 2 phenotypically normal parents. Okay, so that means they're both able to see colour. They produce a colourblind son. A colourblind son must have this genotype. If they're phenotypically normal, that gives us but have a colourblind son, that gives us a clue as to the genotype of the female. She's phenotypically normal because she has this dominant allele on one of her X chromosomes. But she must be a carrier to pass on that X chromosome to her son, so she is heterozygous. Phenotypically normal father would have to have the dominant allele on his X chromosome, and he passes on the Y to his son. So we've received the recessive allele on the X chromosome from the mother, the Y from the father. So construct a Punnett square to show the sex-linked phenotypic ratios. So let's do this in full. So the parental phenotypes. Well, we have a mother who is phenotypically normal, but we know already she is heterozygous, so we can call her a carrier, and I'm using the phrase here, normal. A bit problematic, perhaps. The parental genotypes. Well, the carrier, we've already done this, haven't we? She's got a dominant allele on one X chromosome and a recessive allele on the other. The male, got that up in the corner there, has a dominant allele on his X chromosome and a Y. The gametes produced, so there would be either dominant allele on the X for the female or the recessive allele. The male can either produce the dominant on the X or the Y. if you produce a Punnett square of that, so we've got so a female with 2 dominant alleles, 1 on each of the X chromosomes. A female with 1 dominant allele and 1 recessive allele on her second X chromosome. A male with the dominant allele for normal colour vision, and finally a male with a recessive allele. Therefore, so this is the Punnett square. The phenotypes or the offspring phenotypes would be 1 normal female. You would have 1 carrier female, 1 normal male, and 1 colourblind male. Okay, so it's important to remember here that the perhaps in apostrophes, the normal colour vision is a phenotype, but so is the female. So if you're asked for the offspring phenotypes, if you're asked for the ratio of those, so the ratio is 1 to 1 to 1 to 1. You must include the actual ratio, so the number, as well as the phenotypes of normal and female. Carrier is a phenotype, female is a phenotype, normal is a phenotype, male is a phenotype, colourblindness and male, both phenotypes. Include all of that in your responses. If you can't read my handwriting there, perhaps this is an easier way to see it. Okay, so we can still see each of the genotypes of all of the offspring and their phenotypes above. The probability of having a male with the condition, or male with the condition, obviously here, is 1/4. And in fact, for all of those phenotypes, the probability would be 1/4. So let's have another look. So genetic diagram to show a cross between a colourblind male. Now we know a colourblind male must have the recessive allele on his X chromosome. He must also have a Y chromosome. And a normal female. So normal here rather than carrier, so she must be XB, XB. So the gametes would contain, for the male, a recessive allele on the X chromosome and a Y, 2 dominant alleles on the X's for the female. Ooh. Oh my goodness. Not quite sure why I'm writing on an angle here, but anyway. So we would have 1 carrier female, we would have a normal male, and we would have a second carrier female, and finally a second normal male. So 50 of carrier female to normal male. It's not possible for there to be any colourblind males because the males can only receive either that or that, you know, the dominant allele on the X chromosome from their mothers. It's not possible for the females to show colour blindness because the females must inherit 2 recessive alleles, so 1 on each of their X chromosomes. So the likelihood that a female shows a recessive sex-linked condition is much lower. There it is, a bit neater. Essentially the same thing. Okay, another example, haemophilia. So haemophilia is a condition that leads to loss of blood via internal bleeding when due to the absence of or an incorrectly folded blood clotting factor, either blood clotting factor number 8 or blood clotting factor number 9. Very rare, both of these conditions are rare. It's due to a mutation. So the mutation in the DNA base sequence leads to a change in the mRNA base sequence. Let's go through this. So mutation in the DNA changes the mRNA. That causes a change to the sequence of amino acids. A bit of space down here. That changes the positions of hydrogen bonds or ionic bonds, causing a change to the tertiary structure. Okay, I know that's a little bit of a sideline, a bit of DNA revision, but it's always useful to remember this. Okay, so haemophilia is caused by a recessive allele on an X chromosome. Very similar pattern of inheritance, exactly the same in fact, to the red-green colour blindness. There it is. Haemophilia is a gene on the X chromosome, just down here. There's its loci on the X chromosome, and it's present on that non-homologous region of the X chromosome. So it's not present on the Y chromosome. Men only need to have 1 copy of that recessive allele, let's call it H, on their X chromosome to show the condition of haemophilia. Okay, the pattern of inheritance is precisely the same as that one shown in with colour blindness, and you've got some examples of that in your question booklet, but also in the notes here. So if we just have a look at the notes, you can see in the notes genetic cross. So a carrier female, she has 1 dominant allele and 1 recessive allele on each of her X chromosomes, and a normal male. The gametes, as we showed before, Punnett square, or the genetic cross diagram, shows we'd have 1 normal female, 1 carrier female, 1 normal male, and 1 haemophiliac male. Okay. So there are some little hints and tips for sex linkage, and as I said, recently the questions involving inheritance and genetic cross diagrams have nearly always involved some form of sex linkage. So typically you might have a question something like this: a male something or other are more likely than a female something or other to show a phenotype produced by a recessive allele on the X chromosome. So in the stem of the question, type of things that we're looking for here, okay, we know it's allele on the X chromosome. If the male is more likely, we can assume then therefore that the allele for that condition is recessive. It tells us it's recessive there as well. Okay, so if we explain why that is, here are the 2 marking points. These have come up a lot. I've mentioned it already today, but you must include these 2 marking points. This would usually be a 2-mark question. So females have to inherit 2 recessive alleles. Learn that phrase verbatim, off by heart, write it in, there's 1 mark. Whereas males only have to inherit 1. So for females to inherit 2 recessive alleles, males only have to inherit 1. That explains why it's more likely in a male than a female. Okay, other little hints and tips. There are some in your notes here on this. There are no known examples of genes carried only on the Y chromosome in humans. So if a gene is sex-linked, it can be assumed to be X-linked. We've mentioned that already. Males have a Y chromosome. This could only have come from their fathers. The X chromosome must therefore come from their mothers. This means that man cannot pass on a sex-linked trait to his son. If somehow you have drawn a genetic cross diagram and somehow the male has passed on the recessive sex-linked condition to his son, you've gone wrong somewhere. Have another look. So male children must inherit their X-linked chromosome from their mother. Therefore, X-linked genes are guaranteed to be passed on from mother to son. We've just mentioned this. Recessive sex-linked traits tend to be most common in males. This is because there are no other allele on the Y chromosome, so recessive allele is always expressed. Females can show that recessive phenotype, but they would have to inherit 2 recessive alleles. Back to that, females have to inherit 2 recessive alleles, males only have to inherit 1. Okay, so with haemophilia, here are all the possible phenotypes. 2 X's, 2 dominant alleles in each one, normal. Heterozygous, 2 X's, 1 dominant, 1 recessive, is a carrier. Haemophiliac female would have to have 2 recessive alleles on her X chromosomes. Normal male, 1 X chromosome, which carries the dominant allele. It's normal. Haemophiliac male must have 1 X chromosome carrying that recessive allele. Okay. Really important, another tip that's not mentioned here but is vital for the examiners, is you must show the X and or Y chromosome with the allele in superscript. Remember, the allele is only ever going to be present on the X, so it's either well, depending on what the symbol is. It's either going to be a dominant allele or a recessive allele, but you must show, it's really important to show the chromosome as well as the allele in a sex-linked inheritance. If you don't, you don't get the marks. Some other examples then. This is one with sex linkage and codominance. So if you need to revise codominance, go back to one of my previous videos. Codominance, so both alleles expressed in the phenotype. It means that no allele is dominant over another. Okay, so in cats can be black or they can be ginger. Okay, so this is a female black cat. She has two B alleles on each of her X chromosomes. A ginger female cat will have two X chromosomes and a dominant G on each one. A female who is heterozygous for the black and ginger alleles is what we call tortoiseshell. Okay. Now, actually, there's a little bit more to it than this. There is a phenomenon called bar bodies. You can look it up if you want. Where, if you have a look at a tortoiseshell cat, you can see actually this part of the cat is black. Okay, so in that part of the cat, the black allele is expressed. In this part of the cat, it's ginger. Here, it's the ginger allele is expressed. Here, the black allele. Okay, and that reason for that is very early on in development. The cells activate one of the alleles, or in fact, they inactivate the other allele. So here, the G allele for ginger has been inactivated. Here, the allele for B has been inactivated. If you want to have a look at that, there's a link at the end of this PowerPoint that I'll save in the on the OneDrive. But you can also look up this thing called bar bodies, and that's shown in a few different places. But this is extra. You don't need to know it for the A level. One thing you might want to know: can you get a tortoiseshell male? Good question. So, a male obviously only has one X chromosome. So no, it would not be possible for a male to be tortoiseshell because their X chromosome is either the B allele or the G allele, but not both. But in biology, there is always an exception to the rule. Here is a male tortoiseshell cat. If you want to look this up, you can do. You might already be able to predict. But in fact, this cat has an extra chromosome. Okay. So she is XXY. Or he is XXY. Anyway, there's something for you to go and look up. There is a link to this in the PowerPoint. The PowerPoint will be saved on the OneDrive. If you have any other questions about any of this, just get back to me. I hope it's helpful.